Imagine how difficult it was to study proteins in cells 100 years ago. Magnification could only provide a blurry view of macromolecules. Now we can see all the details of life coming into sharp focus. This will transform medicine and the course of humanity.
So if you want to understand how a car works, you open the hood and you look at the engine. If you want to understand how a protein works, you need to use structural biology to image it.
Structural biology is a field of studies of proteins, how they build, how they function, and how they interact with the other proteins outside of the cell and inside of the cell.
Proteins communicate, but when the communication breaks down, it can lead to a wide range of diseases and disorders. By unraveling the mysteries of how protein molecules within cells work, scientists can design drugs to combat disease. Traditionally, the technique of choice for determining protein structures was X-ray crystallography.
The problem with X-ray crystallography is that you had to obtain crystals of everything, and also it's limited to proteins that are ordered and can be crystallized.
And it was very tough. Sometimes you would wait for crystallization a year, sometimes it never happens. And so it was sort of hindering of progress of structural biology. So Beckman Foundation wanted to establish centers to use cryo-electron microscopy to understand proteins at atomic level. And together with the University of Pennsylvania, we established the center.
Thanks to cryo-EM, we can now study these proteins like never before, which will change everything we know about how our bodies work and how to treat disease.
The Beckman Center for Cryo-EM, its core mission is to provide research labs at Penn and collaborators from neighboring institutions with fast and convenient access to high-quality data which they need to solve the structure of biological molecules. So this is our Titan Krios. It's a 300 kV electron microscope with a direct electron detector, and it is the only microscope of its kind at Penn, allowing people to do things at Penn that they previously had to travel to other places to do.
Cryo-EM allows you to look at big, huge complexes.
Looking at the detailed structure at highest resolution directly inside the cell.
We used to spend many years to design the structure, but now the process is 10 times faster than before.
It has been an absolute revolution.
Right now we have between 30 to 50 users of our microscopes, and all these labs here only scratching the surface.
We study how RNA is synthesized from DNA.
My lab studies a system called the actin cytoskeleton.
My laboratory studies metabolism and epigenetics.
My team studies structural biology directly inside cells.
We have several labs that work on proteins that are involved in cancer biology to further understand how to stop the disease.
And all of these innovations are happening right here at Penn.
This is cryo-EM.
This is the future of drug discovery.
This is the Beckman Center
for cryo-electron microscopy.
Bringing the language of life
into focus.